I'm Sam Woessner, and I was new to Berkshire School this year as a junior. When I was introduced to AMSR, Dr. Birch showed me showed me this sort of like theory for continuing a project about UCHL1 and concussions. I was immediately intrigued because concussions sort of relate to sports, and I really enjoy sports. And so I sort of just ran with this project. I was also introduced to the previous work of two students that also worked with UCHL1, and so that sort of got me going. Um, so obviously, concussions are a growing issue in the United States, especially in youth sports. So finding a biomarker for concussions would make diagnosing a concussion a lot easier and more efficient. Um, so UCHL1 is a protein present in the brain that ubiquitizes or marks other proteins for degradation, so it groups proteins and then they're degraded. So UCHL1 just marks those proteins and then they're grouped and then they're degraded. Um, and then it's theorized and mostly proven that UCHL1 is released into the bloodstream after a traumatic brain injury or concussive episode. And then so my goal of the experiment was to confirm that, and then as well as compare them to um, the effects of microwave exposure on the brain, and if UCHL1 can be a biomarker for both cases. So for the concussion process, flies were placed in a little tube connected to a industrial spring that was pulled back and just flung forward. Flies were hit twice a minute apart, and then left 24 hours for their body to react to the episode, and then frozen. For the microwave, they were exposed to microwaves for about an hour, and then let sit for 24 hours, and then frozen. For the sampling process, um, we had to mash up and sort of degrade these frozen flies, get them all into one homogeneous liquid that can then be analyzed for proteins. The figure here is just is the BSA plot, the standard curve to help determine the concentration of proteins in each sample of flies, the microwave, concussion, and control flies, which are left alone. Um, the gel electrophoresis and Western blot was used to stain the paper, and also was used once we had the Western blot, we could use the gel to stain the paper using this machine down here. And then our results came out as such in the top right. Round one, we had some issues where we figured the proteins were too concentrated, that it was too dark to see any real differences. So in round two, we figured we should lower the concentration by diluting the samples more. And as you can see, it was sort of too diluted. So for round three, we kind of picked in between. But the process was messed up, and during the um, antibody stage, which the antibodies attach to proteins that are present, so they show up on the paper. So they come through on the paper, otherwise you wouldn't see anything. So um, yeah, and so that process was messed up, and so we just see a whole bunch of proteins marked everywhere, scattered, and it isn't very helpful. But besides not reaching a conclusive end, there is a lot of promising results, and because we do see proteins and small differences that just we don't know what they are yet. But there is promise to this experiment, and we do hope that we can reach a conclusive result in the fall or winter of next year. Um, I'd like to thank Dr. Birch and Berkshire School for the opportunity to study this and do this project, as well as the other students who previously worked with UCHO1, Sophie Needles and Michael Schiffer. I had a lot of fun doing this. AMSR provided a lot of opportunities that I think I wouldn't have had almost anywhere else. So thank you. And yeah,